Hello everyone, welcome to this channel, and today we are gonna discuss the mind map of a chapter called biological classification. This is second chapter of our class 11th NCERT textbook in biology, and this video is going to be purely in English since I'm making this video for the students who do not know Hindi. A Hindi version of this video is also available on YouTube on channel NEET Guru, so you can see the Hindi version if you want, uh, if you speak Hindi. Okay. But today's video, in this video, we will discuss this chapter purely in English only. Okay. So the name of our chapter is called biological classification. So we are dealing with classification. What is classification? Basically grouping. You know, grouping or making groups. है ना? Sorry. In this video, you might hear some Hindi from me because that's my like habit speaking Hindi. So ignore it. Okay. Huh. So let's come back to the chapter. The chapter is called biological classification, and I am saying that classification is basically making groups. Okay, we make different type of groups on basis of some criteria, and the organisms which are like plants and animals, we divide them into different groups on the basis of some features that we can see in the plants and animals. Okay, so this is basically classification, making different groups on some or other criteria. Okay, so the first thing we are going to learn in this chapter is systems of classification. Now there are many types of classification which are different, which are based on different type of criteria or different type of characters. Okay, for example, the first scientific attempt to do classification was done by Aristotle. So Aristotle was the first scientist to make a scientific effort towards classification. Okay, before Aristotle, there was classification also before Aristotle started, but those classification were not on the scientific basis. Okay, they were on the basis of use of plants and animals. Okay, so they were not scientific classification. The first scientific classification is done by Aristotle. Now Aristotle divided plants and animals. He divided plants into three categories: herbs, which are like smallest types of plant, then is there is shrubs, and then is trees, which are the largest. Okay, herbs, shrubs and trees. So you can say that this classification is based on height. Okay, Aristotle divided plants on the base of their height and stem quality. Okay, now Aristotle divided animals on the basis of the color of their blood. Though those who have red color blood are grouped in one category, and those do not have red blood, like they have any color blood, like except red, they have white blood, they have yellow blood, or whatever blood they have, they are grouped into other category. So this is the classification given by Aristotle, the first scientific attempt to classify. Plants are divided on the basis of their height, and animals are divided on the basis of the uh, color of their blood, है ना? Color of their blood. Okay, so this was the first scientific attempt to classify. Now let's come to a more advanced classification, which is called two kingdom classification. Now this classification system was given by scientist called Carolus Linnaeus. He also gave a binomial nomenclature. You also have learned in the previous chapter that Linnaeus also gave binomial nomenclature, and Linnaeus also gave two kingdom classification system. Now which two kingdom? The Plantae and Animalia. These are the two kingdoms that Linnaeus made. Now there are many drawbacks in this system of Linnaeus. For example, he didn't separate the organism which are eukaryote and prokaryote. So there are two types of cells, है ना? Eukaryote and prokaryote. These are two types of cells which you will study in cell the unit of life chapter. Okay, so these are two type of cells. The main difference between these cells is the nucleus. The let's like make it smaller. Okay, the eukaryotic cells have a defined nucleus. Okay, and prokaryotic cell does not have a defined nucleus because they do not have nuclear membrane. Okay, this is the difference between eukaryotic and prokaryotic cell. Prokaryotic cells do not have nuclear membrane. So I'm writing here so you can understand a bit that prokaryotic cells don't have cell membrane, sorry, nuclear membrane. Okay, not cell membrane, nuclear, that is nucleus. They do not have a membrane on nucleus. membrane. Okay, so prokaryotic cells don't have nuclear membrane. However, eukaryotic cells have nuclear membrane. Now this is a very basic and very deep difference that is found between organisms, but in the classification given by Linnaeus, that is two kingdom classification, there is no separation of eukaryote and prokaryote. This is the one problem. Now second is, he did not separate unicellular and multicellular organism. Unicellular means the organism have single cell, and multicellular means the organism have many cells. So the amoeba and many other organisms which are multicellular are kept together. Now this is not acceptable, since it is a very basic and important difference. Okay. Now third is, he did not separate photosynthetic and non-photosynthetic organisms. So the plants and animals, the fungi can be seen with algae. However, fungi is generally not photosynthetic. However, algae is generally photosynthetic. So Linnaeus did not separate photosynthetic and non-photosynthetic organisms. Photosynthetic meaning the organism which can do photosynthesis, like plants. Non-photosynthetic means the organism which cannot do photosynthesis, like animals. Okay, so these are the problems that persisted in two kingdom classification. Okay, now let's come to five kingdom classification system. You can see the five kingdom classification a little bit. There is okay, you will say, okay, I will tell you. So third type of classification is five kingdom classification. Now this is the classification which we are going to study throughout the whole chapter, and not even this chapter. We are going to study five kingdom classification in the upcoming two chapters also, that is plant kingdom and animal kingdom. In these chapters also we are going to study five kingdom classification. This classification system was given by R.H. Whittaker. So R.H. Whittaker in 1969 given five kingdom classification on not, and what are the criteria in which he made the five kingdoms. The first criteria is cell structure, the structure of the cell. So on the basis of cell structure, he will separate obviously prokaryote and eukaryote, which was the problem here. You can see that Linnaeus did not separate eukaryote and prokaryote. However, here R.H. Whittaker has taken cell structure as a criteria, so he will separate prokaryote and eukaryote organism. Thallus organization. Thallus organization means body organization. So there are many organisms which are made up of single cell, and there are many organisms which are made up of billions of cells. So using thallus organization, we are going to separate organisms who have one cell, or who have tissue level of organization, who have organ level of organization, or who have organ system level of organization. So this is the second part. And third is nutrition. On the basis of nutrition, R.H. Whittaker will separate photosynthetic and non-photosynthetic. So this problem is also solved. So you can see R.H. Whittaker's classification solves all problem related in given in Linnaeus classification. Okay. So on the basis of nutrition, nutrition means a means of obtaining food is nutrition. Okay. How an organism obtains its food is called nutrition. So on the basis of nutrition, R.H. Whittaker will separate photosynthetic and non-photosynthetic organisms. Okay. So here you can say that five kingdom classification is like more advanced than your two kingdom classification. He also considered modes of reproduction and phylogenetic relationship. What is phylogenetic relationship? Phylogenetic relationship means evolutionary relationship. Okay. So R.H. Whittaker will also consider how different organisms are related according to relationship. है ना? How different organisms are related according to evolution. Okay, which organism came first, which is more primitive and which is more advanced, or you can say which is more old and which is new. Okay, so these are the five things on basis of which R.H. Whittaker did the classification, gave five kingdom classification. Now what are the five kingdoms which we are going to study? So let me quickly write here the five kingdoms which we are going to study. Okay, Monera, this is our kingdom first, which contains bacteria. Then there is Protista, which we will going to study in this chapter, and there is fungi, Plantae, and Animalia. These are the five kingdoms which we are going to study. Now remember that in this chapter we are going to study Monera, Protista, and fungi only. Okay, in this chapter. Plantae and Animalia have separate chapters on their kingdom. The sorry, the chapter on plantae kingdom is called plant kingdom, and chapter of animalia kingdom is called animal kingdom. So these are given in the separate chapters. In this chapter we are going to study Monera, Protista and fungi, only these three. Okay, now coming to Monera. Now we have seen three type of systems of classification. Okay, Aristotle's system, two kingdom classification and five kingdom. Now we are going to study Monera, or our first kingdom. So as I said, in kingdom Monera, the sole member is bacteria. Sole member means only member. So in Monera, you will find only bacteria, different type of bacteria, but there are bacteria only in Monera. Okay, there are no other organisms, only bacteria. So kingdom Monera is dedicated to bacteria. Okay, now bacteria show most extensive metabolic diversity. That is means this means that bacteria as a group, like if we take 100 or 200 different types of bacteria, we will see there are thousands type of metabolic reactions going on in their body. So that's why we say they show most extensive metabolic diversity. Here extensive means you can say many types of, okay, they can say they show different types of reactions in their body as a group. Okay, not single bacteria, we are not talking about single bacteria, we are talking about a group of bacteria. So bacteria as a group, they show different like hundreds and thousand different type of metabolic reactions. So we can say that they show most extensive metabolic diversity. Now second thing is shape. On the basis of shape, there are four type of bacteria: coccus, which is spherical shape; bacilli, which is rod shape; spirilla is spiral shaped bacteria; and vibrio are comma shaped bacteria. So on the basis of shape, these are four types of bacteria which you have to learn. Okay, now coming to heterotrophic bacteria. Now what is heterotrophic meaning? Heterotrophic means the organism is dependent on someone for food. He cannot make he or her or it cannot make its own food. Okay, like humans are heterotrophic, so we are dependent on plants for food, we cannot make our own food. Now heterotrophic bacteria are most abundant, most found bacteria in the world. So most of the bacteria that are found in the world are heterotrophic, so we can say that they are most abundant. They are important decomposers, so they decompose things, so they feed on dead and decaying matters, and they fuse the matter with the soil, you can say. Okay, English is not my first language, so you might find my English pretty wrong at the different times. Okay, but you will understand the chapter, so that's a promise. Okay, so heterotrophic bacteria, they are important decomposer, or they feed on dead and decaying matter. Okay, and they cause many diseases like cholera, typhoid, tetanus, and citrus canker. Citrus canker is a disease which is caused in plants. Okay, and cholera, typhoid, tetanus, obviously these diseases happens in animals like in us. Now coming here, types of bacteria. There are two type of bacteria: eubacteria and archaebacteria. Now eubacteria is called true bacteria. This is basically the true form of bacteria that we know about, eubacteria. Example, there is cyanobacteria. Cyanobacteria, there is two things you need to know about cyanobacteria. The first is their colonies are surrounded by gelatinous sheath. Gelatin is a substance, is a like you can say protective substance. Okay, so cyanobacteria lives in colony, like this is one cyanobacteria, this is other, this is third. Okay, 4 5 6, this is a colony of cyanobacteria. In this there are 6 different cyanobacteria. Now on these cyanobacteria there will be a sheath of gelatin which provide them protection. This is the sheath of gelatin. So this is what we are saying here, that colonies are surrounded by gelatinous sheath. Okay, now second point, what is nostoc and amoeba? These are the two different cyanobacteria. Basically you have to learn the example. If in exam you are asked the example of cyanobacteria, then you should know that nostoc and amoeba, these are two type of cyanobacteria. Okay, now nostoc and amoeba have a special type of cell which called heterocyst. These have special type of cells which are called heterocyst, and heterocyst, the work of heterocyst is nitrogen fixation. Heterocyst does nitrogen fixation. Now those who don't know what is nitrogen fixation, This is basically very simple process. uh, just give me a second. Nitrogen fixation is basically this: take nitrogen from the atmosphere and convert it into ammonia, NH3. This is what we called as nitrogen fixation in short. Okay, so nitrogen fixation is done by which type of cells? Heterocyst are special type cells which do nitrogen fixation. This is one thing you are also have to learn. Okay, so this is all about eubacteria. Now coming to archaebacteria. Now archaebacteria are very old bacteria, like very millions of years old bacteria, and they can survive very harsh habitat, very extreme habitats they can survive. Why? Due to a different cell wall, because they have a very tough, very resistant cell wall. So they can survive in many harsh habitats, many extreme habitats. For example, they can survive in salty areas which have very high concentration of salt, and those archaebacteria that live in salty areas are called halophiles. Just a second. Okay, the archaebacteria that live in salty areas are called halophiles. Now they can also survive in hot spring where the temperature of the water is about 100 degrees Celsius, and the surviving bacteria in hot springs are called thermoacidophiles. So thermoacidophiles are basically archaebacteria which can survive in hot springs. And marshy areas, so there is marshy condition, you have seen in Hindi it is called daldal, है ना? So in marshy areas they can also survive in marshy areas. For example, methanogen is very important because it produces methane, and methane makes basically more than 70% of biogas, or you can say biogas is also commonly called gobar gas. So the production of gas the production of methane in the biogas or gobar gas is basically due to methanogen, which are found in marshy areas, or in methanogen also found in dung of the cattle. Okay, like dung of the cow and buffalo. They are also found in dungs of the cattle and produce methane. Okay, so this is all about bacteria. And one thing, there is one bacteria remaining here, mycoplasma. Okay, you might see this is a little blurry. Okay, this is little blur. Okay, mycoplasma. This is one more bacteria we have to study separately. Reason is this is special. Mycoplasma do not have any cell wall, no cell wall. However, all bacteria have cell wall except mycoplasma. Mycoplasma is the only bacteria that does not have any cell wall. Second thing is, it is smallest living cell. Mycoplasma, the size of mycoplasma, you have to like learn this: 0.3 micrometer. 0.3 micrometer is the size of mycoplasma, and is the smallest living cell that we have seen till today. The third point about mycoplasma which is important: they can survive without oxygen. So without oxygen they can live. Mycoplasma can live without oxygen. They don't need oxygen. These are the three important points about mycoplasma, and this is the reason why we are studying it separately, because all bacteria have cell wall but mycoplasma don't. Okay, this is the smallest living cell we have seen and can survive without oxygen. This is one more bacteria. And here comes our second kingdom, protista. So first we have completed our first kingdom, and now we come to our second kingdom. Let's zoom a little bit. Okay, so coming to protista, this is our second kingdom. Huh? Now protista contains all unicellular eukaryotes. So all organisms inside protista are, first thing is they are unicellular, so they are made of only one cell, and they are eukaryotes, so their cells will have definite they have definite nuclear membrane. Okay, nuclear membrane present. Okay, they have a well defined nuclear membrane. So all unicellular eukaryotes are placed in protista. Now second thing is, boundaries of this kingdom are not well defined. What does this mean? Actually in protista there are many organisms that you will see, they are like kind of mixed, like in the two kingdom classification of Linnaeus, you have said that he mixed unicellular and multicellular and photosynthetic and non-photosynthetic. However, Protista does not have prokaryote, so eukaryote and prokaryote are not together. But Protista have unicellular as well as multicellular organisms. Protista have photosynthetic as well as non-photosynthetic. So all type of organisms you can see on Protista. Only thing that Protista defined is that unicellular organism and eukaryotic organism. Other than this, the organism can be multicellular and unicellular. The organism can be photosynthetic as well as not photosynthetic. That is why we say that boundaries of this kingdom are not well defined. We cannot confine this kingdom to a certain criteria. Okay. Now there are 5 classes under Protista which we have to study: chrysophyte, dinoflagellate, euglenoids, slime moths, and protozoans. These are 5 types of Protista we have to study. Coming to the first, we have chrysophytes. Okay, coming to the first, chrysophytes. Chrysophytes have 2 types of organisms: desmids and diatoms. Desmids are also called golden algae. Only this is the only one thing that you have to learn about desmid. Except this, there is anything that you have to learn about desmid. The only thing you have to learn about desmid is that it is called as golden algae. Coming to diatoms, you have to learn many things about diatoms. For example, diatoms are called chief producer of ocean. This means that they are main producer of essence, so they must produce something. Now let's see what they produce. So the cell wall of diatoms, it is like soap box like. So you have seen the soap box in your bathroom. The cell wall structure of the diatoms are just like soap box. So this you can say it is kind of like this. There is one upper covering and there is one below covering. So this is kind of a structure of the cell wall of diatoms. Okay, so diatom cell wall is like soap box, and have silica which makes it indestructible. Indestructible means that it cannot be destroyed. So cell wall of diatoms cannot be destroyed. And why it cannot be destroyed? Because it have silica deposition. Silica is a very resistant material. Basically, you can say silica is kind of soil. Now you cannot destroy soil. You can like burn it, you can whatever you do, soil does not destroy us. Okay, so cell wall of diatoms have silica deposition and which makes it indestructible. So that is, it cannot be destroyed. Since it cannot be destroyed, it gets deposited over billions of years wherever the diatoms live. Okay, diatoms live in the ocean. In ocean, at the habitat of diatoms, that is wherever the diatoms live, the cell wall keeps getting deposited, it keeps getting accumulated over billions of years, over many billions of years, and this deposition is called as diatomaceous earth. Now when we say that diatoms are chief producer of oceans, what they produce? They produce diatomaceous earth. This is the product that they produce, and this is why they are called chief producer of ocean. Now diatomaceous earth is gritty, and it there are uses of it. Now what is meant by gritty? That it is not smooth. Okay, it is not smooth. And what are the uses? The first use is polishing. It is used in polishing. Second is it used to filter the oils, and it is also used in syrups. Okay, when you have syrup, you can feel the grittiness. You can feel that generally most of the syrups are not smooth. Okay, especially the cough syrup that you uh, have. Okay, they are not smooth, so they are also used in syrup. Diatomaceous is also used in syrup. So these are the 3 uses you have to learn: polishing, filtration of oils, and syrup. This is the only things that you have to learn about chrysophytes. So first class chrysophyte is end, and now we come to dinoflagellates, our second class. So dinoflagellates, we are on dinoflagellates. Dinoflagellates may appear yellow, green, brown, blue, or red due to pigments present in them. So whatever kind of pigment present in the dinoflagellates, its color will be according to that pigment. Okay, for example, the chlorophyll present in the plant gives the green color of the plant. So pigment decide the color of the organism. Here whatever pigments are present in the dinoflagellates, their color will be according to them: yellow, green, blue, brown, or red. Okay, since name suggests that the dinoflagellates. Okay, di means 2 and flagellates matlab flagella. So मैं मतलब बोल रहा हूँ sorry, Di means two and flagellates means flagella. So dinoflagellates have two flagella. Two flagella. Okay, one is longitudinal and one is transverse in a furrow between wall plates. What is wall plates? Basically cell wall plates. Okay, the plates of their cell wall. So dinoflagellates have two flagella. One is longitudinal. Acha, the word longitudinal means along the body. One is along the body. Second is transverse. Transverse means something this. Let me show you the arrangement of flagella and dinoflagellates right here. Okay, let's see that this is a dinoflagellates. Now one flagella will be like this. This is one flagella, and second flagella will be something like this. Okay, one is longitudinal, which is along the body, a second is transverse, which is across the body, you can say. Okay, these are two flagella present in the dinoflagellates. Okay, now dinoflagellates also produce red tides. What are red tides? Due to rapid multiplication of red dinoflagellates. For example, goniologs is a red colored dinoflagellates. When goniologs multiplies rapidly, multiplies means does reproduction. It reproduces very quickly. It releases toxin that can kill fishes. These toxins can kill fishes, and since they are increasing in number, that part of the sea in which they are found appears to be red, and which called red tides. We call them as red tides. Okay, since red dinoflagellates are multiplying and they are growing in number, so that part of the sea becomes red, and that's why we call that it is red tide. So this is all about dinoflagellates. Now coming to euglonates. Euglonates have pellicle instead of cell wall. So cell wall is absent here. What is present instead of cell wall? Pellicle. Pellicle is a protein-rich layer, and it makes their body flexible. Body of euglonates are flexible due to this protein-rich layer called pellicle. Okay. They also have two flagella, similar to dinoflagellates. They also have two flagella. One flagella is long and one is short. Okay. Pigments are found which are identical to higher plants. Higher plants mean like the trees that we see on basis daily life, like the mango or banyan tree. The pigments of euglonates are identical to higher plants. They are similar to higher plants. Okay. Now, if light is present, euglonates are photosynthetic, that they make their own food. But if light is absent, sunlight is absent, then euglonate become heterotrophic, that is they kill some other organisms that feed on them, they become heterotrophic. Okay, so here you can say euglonates can be photosynthetic and non-photosynthetic both, and that's why we have said that kingdom of boundaries of this kingdom is not well defined. Coming to the fourth one, slime moths. Slime moths are saprophytic. Saprophytic meaning they feed on dead and decaying, feed on dead matter or decaying matter. Okay, so saprophytic, they are saprophytic, they feed on dead or decaying matter. Okay, okay, in favorable conditions. Favorable conditions means the condition is good for slime moth. If the condition is good for slime moth, they form an aggregate, or you can say they form a group which is called as plasmodium. So plasmodium here is a group of slime moths when they have there is favorable conditions. Okay, in unfavorable conditions, that is when conditions become adverse or bad conditions, plasmodium differentiate. Plasmodiums basically you can say it uh, it differentiate here means टूट जाता है ना, you can say it breaks. Okay, breaks down. Plasmodium differentiate, or you can say plasmodium breaks down and form fruiting bodies which have spores. Fruiting bodies have spores. So this is the condition in uh, favorable and unfavorable condition about slime moths. Okay, coming to fifth and last, protozoans. Okay, this is last type of protista, which is protozoan. Protozoan is also called as primitive relative of animals. That is, they are primitive means old. They are old relative of animals. They are considered as relative of animal. And there are four type of protozoan. Amoeboid protozoan, as the name suggests, it must be like something like amoeba. Now, amoeboid protozoans have the marine forms have silica shells on their surface. Now, marine forms, those who live in marine water like ocean and seas, the salty water, you can say. Okay, so those who live in marine forms, marine areas like salty water like ocean and seas, they have silica cells on surface. Okay, example, you can easily learn the example because amoeboid example is amoeba and ant amoeba, so both have amoeba in their name. Okay, now coming to flagellated protozoan. Flagellated protozoan, as the name suggests, I am saying naam suggest. Okay, as the name suggests, flagellated protozoans must have flagella. Okay, and there is one flagellated protozoan which is very important, trypanosoma. Trypanosoma is a flagellated protozoan which called sleeping sickness. Okay, so you have to learn that sleeping sickness is caused by trypanosoma, which is a kind of flagellated protozoan. Now, ciliated protozoan. Ciliated protozoan have cilia, as the name suggests, and they have a cavity called gullet. Example, we have paramecium as a ciliated protozoan. Sporozoans, as the name suggests, they have infectious spore-like stage in their life. Example, plasmodium. Now, this plasmodium, which is given in sporozoans, and this plasmodium, which is given in slime moulds, these are different plasmodium. They are not same. Okay. In slime moulds, plasmodium is basically aggregate, or you can say group of mini slime moulds. Okay. In slime moulds, plasmodium is basically group of mini slime moulds. In protozoan, plasmodium is a single, you can say, organism. Okay, so it is a single protozoan. So these are different protozoan. The plasmodium of protozoan and the plasmodium of slime mold, these are different, they are not same. Okay, however name is same. Now this is the second kingdom that we completed. We have already completed Monera. After Monera we completed Protista also, and we are coming to fungi. The PDF of this is also already given in the description. You can click on the description and download the PDF. Okay, now coming to fungi, this is our third kingdom. Out of 5, this is our third kingdom, and only 3 kingdoms we have to study in this chapter. The 4th and 5th kingdom are given in the separate chapter. Now coming to fungi, all fungi are filamentous except yeast. Only one except yeast, all fungi are filamentous, that they are in form of filament, or you can say a thin thread. They are present in form of a thin thread. Okay, so all fungi are filamentous except yeast. Now body of fungi is something like this. This is body, and you can see these are the 3 thread like structure. This thread like structure are called hyphae, and network of hyphae is called mycelium. So what is mycelium? Basically, if you consider all three hyphae, you will say that it is basically mycelium. Okay, so hyphae is single, but mycelium is whole network of hyphae. Okay, now hyphae can be septate or sinusotic. Septate means they have cross walls. For example, this one hyphae, you say there is cross wall and there is many nucleus. Okay, there is one nucleus here, one nucleus here. Let's use the red pen. Okay, you can see there is one nucleus here, one here, one here, one here, and there is cross walls between them. Okay, this is separate septate hyphae, and this one is sinusotic hyphae. Here you can say there are many nucleus, but there is no cross walls, there is no differentiation. Okay, this is sinusotic hyphae. So septae have cross wall, but sinusotic hyphae have no walls, and this is multinucleated. As there is no wall, obviously you can see there are many nucleus, so we can say these are multinucleated hyphae. Okay, cell wall, it is a very important question, and it is asked in NEET many times, and in board exams here. That cell wall of fungi is made up of chitin and polysaccharides. Chitin is very important. You can forget polysaccharide. That will not do much of a difference, but chitin is very important. That fungi cell wall is made up of chitin. Okay. Now there are some diseases like wheat rust, which is called by Puccinia. And there is a special phase called dikaryophase, which is seen in Asco and Basidiomycetes. These are 2 type of fungi. This point, let's skip this point, and we will come back to this point. Okay, we will come back to this point. This is important, and you can't understand this currently. It will take some time. Okay, so let's see. Now let's discuss sexual cycle of fungi. The fungi's sexual cycle have 3 steps: plasmogamy, karyogamy, and meiosis. Plasmogamy is basically protoplasm fusion. Fusion of the protoplasm is called plasmogamy, and fusion of nuclei is called karyogamy. And when protoplasm and nucleus both fuses, the zygote is formed, and then there is meiosis in zygote. Meiosis means reduction division. So after the zygote, after meiosis in zygote, it will give rise to haploid spores. The haploid spores means they have only one set of chromosome. So zygote is 2n. Okay, zygote is 2n type of chromosome, and haploid have n chromosomes, that is half the original number of chromosome. So this is the meiosis or reduction division. Reduction division. So this is sexual cycle of fungi. Having 3 steps, these are the 3 steps you have to learn. Now coming to basis of classification. So fungi are classified into different classes, like you can see Ascomycetes, Basidio, and Deutero or Phycomycetes. 4 type of fungi are there, on which basis we have divided them on the basis of morphology of mycelium. So if their mycelium is septate or aseptate, this is the first thing that we have seen. The second is mode of spore formation, that how the spores are made in the fungi, and third is fruiting body, that is type of the fruiting body. Okay, this is the 3 bases on which we have classified the fungi. The first class of fungi, Phycomycetes, is here. So there are 4 classes we have to study: Phycomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes. First, coming to Phycomycetes. In Phycomycetes, the most important feature is that their mycelium is cenocytic, and only Phycomycetes have cenocytic mycelium. All other mycelium, that is in Ascomycetes, in Basidio, and in Deuteromycetes, mycelium will be septate. The only cenocytic mycelium that is seen in fungi is in Phycomycetes only. So this is a very important point. It's very important point. Only Phycomycetes have cenocytic mycelium. Reproduction. Asexual reproduction is from zoospore and aplanospore, and sexual reproduction is by zygospore. Now, zoospore and aplanospore are endogenously produced. That is, they are produced within the body of Phycomycetes. That's we call endogenously produced. Okay. Examples we have ram. Ram means Rhizopus, albugo, and mucor. So you can run Phyco ram. Phycomycetes have 3 examples: Rhizopus, albugo, and mucor. Okay. Coming to Ascomycetes. Ascomycetes Ascomycetes is called sac fungi. This is a common name given to Ascomycetes. It's called sac fungi. And coming to reproduction, asexual reproduction is by conidia, which is exogenously formed, while sexual reproduction is by ascospores, which are produced endogenously on asci. Now, what is asci? Basically, fruiting bodies. Because you know, I have already taught you that fruiting bodies give rise to spores. So here, ascospores are produced endogenously, that within asci. Okay, asci is here fruiting body, you can say. Okay. Now coming to examples. So there are 3 main examples you have to learn: can, C-A-N, claviceps, aspergillus, and neurospora. Okay, this is the shortcut. Then you have to learn other examples also. Okay, can, and then you have yeast, penicillium, morels, and truffles. These are the examples. So first thing you have to learn can. Can means claviceps, aspergillus, and neurospora. Then you have yeast and penicillium. The penicillium is the one that gives us penicillin, antibiotics. Okay. So penicillin we get from penicillium, and penicillium is which type of fungi? You can say Ascomycetes. Okay. This is the type of question you can see in the NEET or your board exams. Okay. Now, morels and truffles are edible type of fungus. Okay, these are edible, and people eat them. Okay. Now coming to neurospora. Neurospora is important because it is used extensively in genetics work. Though neurospora is widely used, you can say they are widely used in genetic work to study genetics. Okay. So coming to third fungi, Basidiomycetes. Basidiomycetes is also known as bracket fungi or puffball, and these are common name. Okay, bracket fungi or puffball, or you can also found somewhere mushroom written. Okay, mushroom is also commonly called for Basidiomycetes. Coming to reproduction, asexual spores are generally not found. Generally, this is not a compulsion. This is generally. Okay. So in Basidiomycetes, generally asexual spores are not found. Sexual spores are basidiospores, which are produced exogenously on basidiocarp. Exogenously, it means outside the body. They are produced outside the body on a fruiting body, which is called as basidiocarp. Here basidiocarp is basically fruiting body. Now coming to example, you have to learn pua, P-U-A, pua. Puccinia, Astilago, and Agaricus. These are Basidiopua. Okay, Basidio have three examples: Puccinia, Astilago, Agaricus. Now, Puccinia is called as rust fungus, Astilago is called as smut fungus, and Agaricus is basically mushroom. Okay, so mushroom comes in Basidiomycetes. So these are the examples. Coming to fourth and last, Deuteromycetes. Deuteromycetes fungus are called as imperfect fungi. The reason is the sexual reproduction of Deuteromycetes fungus are never known. Okay, so the members of Deuteromycetes do not show sexual reproduction. We do not know sexual stages. Okay, and asexual reproduction in them are is through conidia. Okay, examples we have cat. Cat means Coletotrichum, Alternaria, and Trichoderma. This is the example: cat, Coletotrichum, Alternaria, and Trichoderma of Deuteromycetes. You have to learn that. Now this completes our third kingdom, which is fungi. Okay, we have totally studied three kingdoms. Coming here again, we have studied Monera, Protista, and fungi. Plantae Animalia are given in different chapter, but in this chapter we have to study some acellular organisms, that is viroids, and prions. Okay, coming to virus. Now the topic we are currently discussing, these are called acellular organisms. Acellular organism. Like virus is a acellular organism. Okay, viroid is a acellular organism, and prion is a acellular. These are acellular organisms. And why we are saying acellular? Because the body of virus or viroid or prion is not made up of complete cell. That is why we call it acellular, or cell is absent here. Basically they are not made up of complete cell. That is why they are acellular. And the reason they are not made of complete cell, this is the reason why we say that virus is not truly living. We cannot say virus is living because it does not have a cell of structure. And that's why similarly viroid and prions are also not living. But since they show features of living things, some features, that's why we have to study them. Okay, virus. Coming to virus, not truly living. Okay, discovery. Dmitry Ivanovsky was a scientist. 1892. In 1892, he discovered virus, and he discovered about tobacco mosaic virus. Actually, he was studying about tobacco roots. I think I'm not confirmed, but he was studying about tobacco roots, in which he found tobacco mosaic virus. Short form में, sorry, we say TMV in short form. Okay. The genetic material of this virus is RNA, and it has a protein coat called as capsid. Now capsid is a coat which is made up of many units called as capsomere. Okay. Coming to next, M.W. Bejernik is a scientist who called virus as contagium vivum fluidum. Now contagium term means the thing which can be contagious, or you can say which can be infectious. So virus is a infectious. It can spread from one person to other, one organism to other. Okay, so virus is contagium. It can spread from one organism to other. Vivum means this virus is a poison, you can say. Okay, and fluidum means fluid. So M.W. Bejernik basically said that virus is a infectious poisonous fluid. Okay, so this is the meaning of contagium vivum fluidum, and this sentence is already asked in NEET previous year questions, so you have to learn it carefully. Okay, coming to Stanley crystallized virus. So he made crystals of virus. He was the first to make crystals of virus. No virus contains both RNA and DNA. This is a very important point. No virus is seen till today that has both RNA and DNA. Only the viruses can have only RNA or only DNA. Only one of them. They cannot have both RNA and DNA. For example, you have seen there tobacco mosaic virus have RNA only. Okay. Now plant virus, that is the virus which infect plant, is called as plant virus. Plant virus generally have single stranded RNA. Now this is a general term. This does not means compulsion. Okay, it is not compulsory, but generally plant virus have single stranded RNA or ssRNA. Okay. Animal virus, that is virus that infect animals, have single stranded RNA or double stranded RNA or double stranded DNA. These three can be found in animal virus. Bacterial virus means the virus that affects bacteria, that attacks bacteria, is called bacterial virus. Bacterial virus is bacteriophage, basically is a bacterial virus. Bacteriophage is very important. It is basically a virus that attacks bacteria. Bacteriophage is a virus that attacks bacteria, and it has double stranded DNA. Okay, so here comes the viroids. We have done with the virus. Viroid is also a cellular organism. Okay, this does not have a complete cell structure. It is free RNA only. This have only RNA. Virus have genetic material plus protein code. There were two things. That is, any virus can have RNA plus protein or DNA plus protein, but viroids can have only RNA, nothing else. Okay, so viroids are free RNA only. They are smaller than virus, and they are discovered by T.O. Diner in 1971. Now coming to RNA, the viroids RNA is of low molecular weight than virus RNA. Okay, the RNA in virus is of high molecular weight. Okay, and in viroids it is of low molecular weight. This is one thing you have to learn. One disease caused by virus, that is potato spindle tuber disease. Okay, virus diseases caused by virus are commonly known to us. For example, common cold, which is caused by rhinovirus. Okay, and HIV, AIDS is caused by HIV, human immunodeficiency virus. Okay, so these are the diseases by virus which are already known to you, but viral disease is rarely taught. So potato spindle tuber disease, this is caused by viruses. Coming to prions, prions have only protein, abnormally folded protein. So there is the protein is not normal. Okay, it is abnormally folded. So prions have only protein, no RNA, no DNA, only protein. So prions are basically abnormal folded protein. Okay, their size is similar to virus, and there are two diseases you have to learn. BSE, that is bovine spongiform encephalopathy. This is the name of the disease. BSE is full form, bovine spongiform encephalopathy. It is also called mad cow disease. And the similar sorry I mean the disease CR Jacob disease, that is CJD disease, occurs in human, and this disease have similar symptoms like mad cow disease. So mad cow disease, or you can say BSE and CJD, these have similar symptoms. Okay, CJD happens as human, but BSE, bovine spongiform encephalopathy, it happens in cows. Okay, it is mad cow disease, cattle में देखी जाती है. Okay, coming to lichen. Lichen is a symbiotic association. Symbiotic association means an association in which two organisms live together and both are benefited by each other. Okay, so symbiotic association between algae and fungi. So algae and fungi will live together. Algae gives something to fungi. Fungi will give something to algae. Now, what is work of algae? So algae prepare food and it will give food to fungi. Now, what is work of fungi? Fungi will give shelter to the algae, so a place to live, and fungi will absorb water and mineral and give it to the algae to make the food. Okay, now algal component or the algae in a lichen is called basically phycobiont component, and the fungi in the lichen is called mycobiont component. Okay, fungi is mycobiont, algae is phycobiont. Lichens are good pollution indicator. They do not grow in areas which are polluted. Okay, so this was the last line in our mind map, and you can download this mind map from the link given in description of the video. Okay, so this is the chapter of biological classification, and we will meet in the next video. Till then, goodbye. And if you like the video, you can share with your friends, and obviously hit the like button and subscribe to the channel. Okay, and if you have any question related to the video, anything that you do not get or you have a confusion, you can comment. Okay, I will respond in the comment, and I will give you answer in the comment. Okay, so you can always comment, and obviously you will get answer sooner or later. You will get the answer. Anything you have discussion you want to discuss from the chapter, you can comment. Okay, so that was it. We will meet in the next video. Till then, goodbye, have fun.